Hello viewers, you are welcome to my YouTube channel Fasihuddin. This is the third episode on Angiosperm morphology where I will be discussing on the dicot and monocot morphology. Dicotyledons and monocotyledons represent the two major lineages within the Angiosperm, a group characterized by its remarkable diversity and ecological significance. While united by the shared feature of producing flowers, these two groups exhibit striking differences in their overall morphology, reflecting distinct evolutionary pathways and adaptations. This presentation delves into the comparative analysis of the key physical characteristics, aiming to eliminate the fundamental distinctions that define these plant classes. Understanding the morphological traits that differentiate dicots and monocots is crucial not only for taxonomic classification but also for comprehending the broader implications of plant evolution. By scrutinizing the features such as the root system, stem structure, flower morphologies, and pollen characteristics, we gain insight into the functional significance of these variations. This comparative study aims to provide a clarity on the morphological relationship between these two significant groups, revealing the fascinating diversity within the Angiosperm lineage. However, it is important to note that modern classification, particularly with the advent of molecular phylogenetics, has refined our understanding of the Angiosperm relationship. The term eudicots is now used to describe the large majority of what were traditionally considered dicots. But the basic dicot monocot division is still very useful for the study of basic botany. Understanding the classification of the dicotyledons and monocotyledons is a fundamental in botany for several key reasons, impacting both the scientific study and practical applications. These classifications provide a structured framework for organizing and understanding the vast diversity of angiosperms. It allows botanists to group plants with similar characters, making it easier to study their relationship and evolutionary history. Knowing the differences between the dicots and monocots, it aids in plant identification. Many staple food crops are monocots and some are dicots. Different groups have different needs regarding the soil, water, and nutrients. The herbicides often target either monocots or dicots, so proper identification is essential. In breeding programs often focus on specific traits within the groups. Gardeners and the landscapers use this knowledge to select and care for the plants. Understanding the growth pattern, root systems, and other characteristics help for the successful cultivation. Plant communities are often characterized by the dominance of either monocots or dicots. This knowledge is essential for understanding the ecosystem dynamics, conservation efforts, and restoration projects. Many medicinal plants are the dicots. Knowing their classification, it helps in identification and studying their properties. The study of dicot and monocot classification contributes to our understanding of plant evolution and their relationship between the different plant groups. The differences in their structures reflect the adaptations to diverse environments. Studying these differences will help to understand how plants have evolved to thrive in different conditions. Cotyledons are fundamentally the embryonic leaves that form within the seed, serving as the seedling's initial leaf structures. They are among the first parts of the plant to become visible upon germination. A critical role of the cotyledons is to act as nutrient reservoir. They often store food reserves which nourish the germinating embryo until the young plant can produce its own sustenance through photosynthesis. In some species, cotyledons can even perform the photosynthesis themselves, providing the seedling with an early source of energy. Beyond their nutritional function, the number of cotyledons present in the seed serve as the primary characteristic of classifying the flowering plants. The monocotyledons or the monocots are defined by having single cotyledons, while the dicotyledons or dicots are characterized by possessing two cotyledons. The leaf venation refers to the pattern of arrangement of veins within the leaf lamina. These veins are essentially the vascular tissue of the leaf responsible for the transporting water and nutrients through the leaf blade. The pattern of venation is the key characteristic used in plant identification, particularly in distinguishing between the monocots and dicots. They provide a structural support to the leaf. They transport water and minerals from the stem to the leaf. They transport sugars produced during photosynthesis from leaf to other parts of the plant body. In dicotyledons, which exhibit reticulate venation, this means the veins form a network of branching out in various directions, creating a web-like or net-like pattern. Often there is a prominent central vein called the midrib, from which the smaller veins branch out. In monocotyledon, they exhibit parallel venation. This means the veins run parallel to each other along the length of the leaf. These veins may be relatively straight and run from the base of the leaf to the tip of the leaf. The stem structure varies significantly between the dicotyledons and monocotyledons, primarily in the arrangement of their vascular bundles. These differences are crucial for understanding how these plant groups transport water and nutrients and provide structural support. In dicotyledons, the vascular bundles are arranged in the form of a ring around the stem's periphery. Each vascular bundle contains xylem, which transports water and minerals, and phloem, which transports sugar and other organic compounds. And these two are separated by a layer called cambium. The cambium is a layer of actively dividing cells that allows for the secondary growth, leading to the increase in the thickness of the stem. This is why many dicots grow into trees or the shrubs. The center of the dicot stem is often filled with the pith, a spongy tissue. The area between the vascular bundles and the epidermis, that is the outer layer, is called the cortex. This organized ring structure provides strength and flexibility. The cambium allows for the development of woody tissues, enabling for the long-term growth. In case of the dicot monocotyledon, the vascular bundles are scattered throughout the stem's ground tissue. These bundles are composed of xylem and phloem, but they lack cambium layer. Because they lack the cambium, monocots typically do not exhibit secondary growth in the same way as dicots. The ground tissue called parenchyma fills the spaces between the scattered vascular bundles. There is no differentiation between the pith and the cortex. The scattered vascular bundles provide flexibility, which is particularly useful for the plants like grasses that need to withstand wind. The absence of cambium limits the stem's ability to the thickness significantly. The root systems are crucial for the plant survival, anchoring the plant in the soil and absorbing water and nutrients. Dicotyledons and monocotyledons exhibit distinct root system types reflecting their evolutionary adaptations. Dicots typically possesses a taproot system. This system features a prominent thick primary root called the taproot, which grows vertically downward. The smaller lateral roots branch out from the taproot. The taproot penetrates deep into the soil, providing the strong anchorage and access to the deeper water sources. In monocotyledons, they exhibit fibrous root system. The system consists of a dense network of numerous thin branching roots that grow from the stem's base. These roots spread out horizontally, forming a mat-like structure. Fibrous root systems provide good soil stabilization and prevent soil erosion. They are effective in absorbing water and nutrients from the upper soil layers. The structure of the flower parts is another key distinguishing feature between the dicotyledons and monocotyledons. The number and arrangement of the parts often follow the distinct pattern in each group. In dicotyledonous flowers, parts like petals, sepals, stamens, and pistils typically occur in multiples of 4 or 5. For example, a dicot flower might have 4 petals, 4 sepals, 8 stamens, and 1 pistil. The arrangement of these parts can vary widely, but they are often arranged in distinct whorls. The petals and sepals can be distinct and showy. In monocotyledonous flower, the parts are typically occur in multiples of 3. For example, the monocot flower might have 3 petals, 3 sepals, 6 stamens, and 1 pistil. The arrangement is often simpler than the dicots. Often the petals and sepals look very similar and are then called tepals. These differences in flower structure are useful for the plant identification. They also reflect the evolutionary adaptations related to the pollination strategies. The number of flower parts is a reliable visual clue. Pollen structure, particularly the number of apertures present on the pollen grains, serve as the critical distinguishing feature between the dicotyledons and monocotyledons. The dicot pollen grains are typically tricolpate, meaning they possess 3 apertures or furrows, known as colpi. This tricolpate structure is a hallmark of most dicots and plays a vital role in pollen germination, facilitating the emergence of the pollen tube. Conversely, monocot pollen grains are usually monocolpate, characterized by a single aperture or a furrow. Similar to dicot, this aperture is essential for the germination process. The consistent difference is the aperture number. 3 in dicots and 1 in monocots makes the pollen structure a highly reliable characteristic for plant classification, particularly at higher taxonomic levels. Furthermore, pollen ability to be preserved in fossil records renders this characteristic invaluable for tracing the evolutionary relationship between the plant groups, offering insight into the historical development of the flowering plants. Dicotyledons and monocotyledons are the integral to the functioning of the ecosystems and their sustenance of human societies, each contributing uniquely to both ecological balance and economic prosperity. Ecologically, dicotyledons form the structural backbone of the numerous habitats, with the dicot trees providing shelter and resources for the diverse array of animal species. Their taproot system play a vital role in stabilizing soils, preventing erosion, and facilitating water penetration. The immense biodiversity within the dicot group enhances the overall richness of the plant communities, and many dicots are crucial for the pollination supporting the insect populations. Furthermore, dicots like legumes contribute to the soil health through a symbiotic relationship with nitrogen fixing bacteria. In contrast, the monocotyledons, particularly grasses, excel in soil stabilization through their fibrous root system, which effectively prevent erosion and maintain the integrity of the grasslands. They serve as the essential primary producers, forming the foundation of many food webs, and monocots like reeds and sedges are the critical components of wetland ecosystems. Economically, dicotyledons provide a vast array of resources that are indispensable to the human life. Many staple food crops, including fruits, vegetables, and nuts, are dicots and are sources of essential cooking oils like soybean and sunflower. Hardwood trees like valuable timber for construction and furniture or numerous medicinal plants are the dicots, contributing to both traditional and modern medicine. Additionally, dicots provide fibers like cotton and jute. Many monocotyledons, on the other hand, are the sources of some world's most critical staple food, including rice, wheat, and corn, which contribute to the substantial portion of the global calorie intake. Bamboo, a monocot, serve as a sustainable building material and fiber like agave and coir are so harvested from the monocots. Ornamental plants like lilies and orchids enhances the aesthetic value of and the landscape. The sugarcane, a monocot, is a vital sugar producer. Thus, both the dicots and monocots play an inseparable role in supporting the ecological and driving